almost 2:15. Can we have everyone seated, please?
Okay.
Live stream Yes, ma'am. Sir, sir. Started. Yeah, but that's
Kind attention, audience. Can we have all of you seated inside the hall, please? I hope I'm audible. I hope I'm audible to every one of you. Please be seated inside the hall, please.
അതിൽ മറിയയുടെ ലിങ്ക് കോപ്പി
As we have already passed this time,
ഡയറക്റ്റ് നമുക്ക് വാട്സാപ്പിലിട്ട് അയക്കാം.
it's 2:15 right now. Can we all please be seated inside the hall and proceed to the next session?
ഇതിലും കിട്ടട്ടെ യൂട്യൂബിൽ ഇട്ടോ മറിയിക്കും കൂടെ അല്ലെങ്കിൽ ഇതിനകത്ത് മറിയിക്ക് എടുക്കാമല്ലോ. ഓക്കെ അത് ഓക്കെ അല്ലേ സാർ? അതിൽ യൂട്യൂബ് ഓൺ അല്ല, ഇത് സ്ക്രീൻ ഈ രണ്ട് ചെയറെടുത്ത് മാറ്റിക്കേ അങ്ങോട്ടും ഇങ്ങോട്ടും. ഓക്കെ ഓക്കെ
Where a 3C's join at Kanyakumari is a multidisciplinary university with a choice-based credit system. Join University.
An announcement, YouTube live streaming is already being done of the conference, and also we have the Google Meet going on side by side. I invite all the audience who are outside after the lunch to kindly be seated in the hall, and also all the posters to be at the registration counter. And with that, we go to the next session after the lunch break. I hope all of you enjoyed your break and got to know many people out there. And now we go on forward with the plenary session with Dr. Gayatri V, Senior Scientist, Phytochemistry and Phytopharmacology Division, JNTBGR Palod, Trivandrum, on the topic plant-based nanoparticles for advanced medical applications, current trends and future perspectives. I welcome you on stage, madam. Dr. B. Gayatri is the Senior Scientist, Phytochemistry and Phytopharmacology Division, KSCSTC Jawaharlal Nehru Tropical Botanic Garden and Research Institute, Palod, Trivandrum.
Hello. Thank you for the introduction. And I think after the post-lunch siesta, everybody would like to sleep. I'll not bore you much with my slides. Anyhow, thank you for the opportunity. I thank Anu, Managing Director of Celogen, for giving me the opportunity, as well as all the other staff members. Moving on with the slides. My today's topic is plant-based nanoparticles for advanced medical applications, current trends and future prospects. Plant-based nanoparticles, plant-based molecules or the phytochemicals are currently being used in many pharmaceutical companies as well as nutraceuticals. And many, green chemistry is called, and many nanoparticles have been synthesized with the help of plant materials. But rather, they are plant extracts are rather used to reduce the inorganic molecules into nanoparticles rather than using the plant phytochemicals as nanomolecules. Moving on with the slides, a brief introduction about nanoscience, nanotechnology, and other aspects. We know that nanoscience or nanotechnology is the emerging field, and nanoscience deals with the making, manipulating, and concentrating fabrication of the particles or the inorganic molecules of size 1 to 10 nanometers. Whereas coming to an organic molecule, you cannot restrict the size from 1 to 10 nanometers. So a new definition has been coined. It says that it is 1 to 1000 nanometers. And this nanoscience application or the device which is made of nanoparticles is called the nanotechnology. Another aspect is the nanochemistry or the phytochemistry, phytomolecules or the synthetic molecules which are used in chemistry to make a nanoparticle is called nanochemistry. They give a definite shape, size to the molecules, composition, surface structure, charge, and functionality of these molecules lead to the nanoparticles. Coming to our area of expertise, that is phytochemicals, we are a tropical botanic garden and research institute where our lab focuses on the metabolites from the plant sources. So there are basically two metabolites. One is the primary metabolites, another is the secondary metabolites. Primary metabolites of the plants are mainly carbohydrates, proteins. Like human beings, they too have primary and primary metabolites, which comprises of carbohydrates, proteins, lipids, and etc. The secondary metabolites are the metabolites which are not directly involved in the metabolic pathway. And what are these secondary metabolites, or why should the plant have a secondary metabolite? Secondary metabolite are the molecules or the chemicals which help in the protection of the plants from the predators, pathogens, competitors, and help in pollination and help the combat them from the abiotic stress, used in therapeutic medicine, and also as used as additives in the industrial areas. Now, secondary metabolites. Secondary metabolites are synthesized from the basic pathway of the plant metabolism, that is photosynthesis. Once photosynthesis takes place, you get sugars, and these sugars are then go into the pathway to form saponins and cardiac glyocytes. Then it goes to the respiration. This sugar is used for the respiration, as we all know. It enters the acetyl-CoA pathway, where it again gets converted byproduct forms called as terpenoids and sterols. Then phenylyl-CoA, fatty acids, flavonoids, tannins, phenols, alkaloids, lignins, etc. These are all the secondary metabolites of the plant, plant molecules. Over 250,000 secondary metabolites have been identified, and they are classified according to their biosynthetic pathway. As I've already said, they are major alkaloids, glycosides, phenolics, and terpenoids. Now, my today's topic, I'll be focusing on these secondary metabolites which can be used in the treatment or as a therapeutic molecule for the diseases, human diseases. There will be two diseases which we have focused. One is the seizures, another is the brain tumors. Seizure is nothing but the convulsions after the epilepsy has occurred, and brain tumors, as you all know, it's a tumor inside the brain. This is our first publication, which we have worked on the asiaticoside molecule. This is nothing but, many of you know Centella asiatica, which is a prominent elixir of life. It is called. It is a neuroprotectant plant. It's called Centella asiatica. In North, it is called as Brahmi. This plant has two major molecules. One is asiaticoside, another is madagasoside. Already, these molecules are said to possess neuroprotective activity. But the problem with neuroprotection, as we all know, brain has different compartments. It has blood-brain barrier. The crossing of any molecule above a size becomes very difficult for its action. So you have to make the molecule admissible to cross the barrier. So what can you do? You can convert this molecule into a nanoparticle inside an encapsulated polymeric matrix and help to cross or help to get accepted into the receptor of the brain and transported inside the brain for its action. Here we have used asiaticoside, which has been encapsulated inside the chitosan alginate polymeric matrix, and this has been transported inside the seizure model in mice, which was induced with the help of pilocarpine. Pilocarpine is a well-known seizure-inducing molecule. It helps in exciting the excitotoxicity. This is another paper which talks about the becoside 2 nanoparticles in 6 glioma cells. Okay, becoside is another molecule from another plant which has another, neuroprotective action. It is called Bacopa moneri. This is also called Brahmi. All these things you find inside the Chyawanprash.
So these molecules basically they are said to have neuroprotective action, but in vitro. We don't know how far they have been active inside the brain. So what we did, we again made this Vecoside 2 nanoparticles to help in inhibition of the C6 glioma cell. Glioma is nothing but the brain tumor. Now going into the work of this paper, we first isolated the Becopocide 2 from the Bacopa moneri. Becopocide 2 is a jujubinil. It is a terpenoid saponin from Bacopa moneri. This was sequentially extracted and it was again partitioned and column chromatography, and then the pure compound Becoside 2 was obtained. This was characterized with the help of NMR, FTIR and HPTLC, the standard Becoside A to ascertain its purity. Now, after the extraction, after getting the pure molecule, we encapsulated this Becoside 2 into the PLGA PEG polygon matrix. Then this encapsulated with the help of nanoprecipitation and solvent displacement method. 1%, 0.1% gluconic acid was used as a surfactant. It was lyophilized later. After the nanoparticles were formed, we wanted to characterize the nanoparticles, which was done with the help of dynamic light scattering, zeta potential, and transmission electron microscope, XRD. We went for drug loading efficiency and encapsulation efficiency. We also, found the release profile, that is how much Becoside 2 was released from the polymer matrix with the help of dialysis method, and it was quantified with the help of UV spectrophotometer. All these details can be found inside our paper, which is there in Phytomedicine Plus of Elsevier journal. Then coming to the C6 glioma cells, these are the rat glioma cells, brain tumor cells, which were cultured, and positive control was BCMU, which is a drug which is used to treat glioma in patients, and that is the Becopocide 2 nanoparticles was treated for 24 hours into the cell, and cytotoxicity was evaluated. For characterization purpose, NMR was used and FTIR was done to see the encapsulation inside the polymer matrix. XRD, the bottommost figure shows the XRD, which has an amorphous nature of the particle. And DLS, the zeta potential was negative, which helps. Usually, zeta potential gives you an idea about the homogeneity and the transportation or the polydispersity index of the molecule. So, more the negative, the more it is better to cross the barrier. So, it was for us, it was helpful. So, it helped us to, help our particles to cross the blood-brain barrier. The release kinetics. Initially, there was a burst of the particles, and then it became controlled release. Then cytotoxicity in the normal cells, they did not find, and we did not find any cytotoxicity. Whereas for C6 cells, we could find the cytotoxicity. Normal cells were, neuronal stem cells. Then this was the proposed structure of our molecule. The top one is the Becopocide A, which is a complex organic molecule from the plant Bacopa moneri, which was isolated in our lab and which was encapsulated inside the PLGA PEG matrix, and we formed this nano capsule. Now coming to the next assay, we saw that our molecule caused nearly late and early apoptotic in flow cytometer. Compared to the drug control, our molecule had a prominent apoptotic behavior towards the C6 glioma cells. Again I'm saying C6 glioma cells are the brain tumor cells, and in ROS, that is reactive oxygen species, during the growth of the cancer cells, there's always an stress increases inside the cell. So we wanted to know what is the stress level inside the cell.
So we went for reactive oxygen species analysis by flow cytometer. We could see that our particle induced more of the stress inside the cells. Why? Because to make the cells die, it generated more of ROS inside the cells, C6 cells. Topmost slides are the EDU analysis, that is incorporation of the DNA synthesis inside the cells. You can see that the control cells were all healthy and there was no degeneration of the cells. Whereas in the B, you could find there were disintegrated cells in the in the picture. So this denotes that DNA synthesis was reduced when the cells, C6 cells, were treated with the nanoparticle. And bottom C and D gives you an idea about the cell cycle analysis. We found that there was a G2 S1 and G2M phase checkpoint where the synthesis of DNA was restricted. So our particles caused a block or the senescent of the DNA synthesis from the S phase. Then coming to the uptake. So we really wanted to know if our particles or due to any other factor does it happen. See, then we went for a uptake study, that is the particle entry into the cells through flow cytometer, that is SSC and FSC scatter. We could find that our particles reach the cells. Like normal cells, only 30% cells were there in the P3 quadrant, but in treated cell there was 38.2. That is, entry of the particles were more prominent in the P3 quadrant. All the drugs kill the normal cells also. This is what happens in cancer. So when you give a drug, normal cells are also targeted. Why? We don't know the mechanism why it happens. So for that, we wanted to know if our nanoparticles could damage the normal cells. So we isolated the astrocytes from the mice brain and we stained it with GFAP, which is a marker for astrocytes. And we could find that there was an intact astrocytic cytoplasmic extensions which were prominent. There was no degeneration of the astrocytes were seen, or no neurotoxicity was noticed. Conclusion from this study says that this particle is safe and it can be used in treating the brain tumor, which is a very much challenge to neuroscience, and which was achieved by the internalization of the particles inside the C6 cells and increasing the ROS concentration as well as inhibiting the cell proliferation and also inducing the apoptosis in the C6 cells. Coming to another work which we wanted to study on C6 cells with the metacosicide, which is another molecule from the Centella asiatica. We complex the molecule with the polymer matrix, and we saw that its degradation, that is the thermal degradation, and the DSC dynamic, differential scanning, colorimetry, everything was within the range as that of the biocompatible chitosan alginate matrix. FTAR, we could find prominent peaks or the peaks which were very much characteristic of the metacosicide, which were in line with the our nano particle also. In SEM, we could find that the particles were highly porous, and in TEM image also we could find the particles were spherical inside and morphology, which having a size of 400 to 600 nanometers. Then we wanted to check our particles with the human glioblastoma cells, that is U87, and we found that our particles up to a concentration of 200 did not cause any, cause the cytotoxicity, or you can see the extensions of the astrocytes being degenerated, whereas the mother molecule, that is metacosicide, did not cause any toxicity to the cells. Similarly, apoptosis also we noticed that particles, that nanoparticles were more apoptotic compared to the control cells. Also, in DNA condensation method, we could see blebbing and nuclear condensation of the DNA inside the C6 cells with the help of staining, and acridine orange staining also proved this, stating that the cells were undergoing apoptosis followed by necrosis. Similarly, this was also substantiated with the help of ROS protection, where we could find there was increase in the reactive oxygen species in the cells. P2 quadrant, there was an increase in the intensity of the fluorescence signaling, and again the EDO incorporation of DNA synthesis and the cell cycle analysis both proved that there was there was a crunch in the DNA synthesis. Again, uptake study, we could find there was a transport of the molecules inside the cells, which helped in the control of the C6 cell proliferation. Another study where methotrexate was used as an to treat the excitotoxicity in the pilocarpine-induced seizures. This is a schematic diagram where how we have made the nanomolecules or colloidal suspension, and it was physicochemically characterized with the help of DLS, zeta, SEM, TEM, and XRD, and release was found. Size was analyzed with the help of DLS spectrum. Zeta potential gave a negative zeta potential, and the surface morphology was porous, and TEM gave a spherical morphology followed by amorphous nature of the molecule, and a release, we could get a sustained and controlled release of the drug. This was given to the rodent pilocarpine-induced seizure rodent model. Seizure is nothing but epilepsy. We have induced epilepsy inside the mice model, and we could found that the intensity of the seizure, the plus sign denotes the intensity. The more the plus sign, the more the intense or more the seizure or more the epilepsy is. So with the help of, when you give the pilocarpine together with the molecule, the intensity of the seizure decreases. Similarly, positive control was levetiracetam, which is a drug which is used to treat epilepsy. Our molecule also had a beneficial effect in reducing or in controlling the epilepsy. And we wanted to test the cytotoxicity of our molecule in the normal cells. Normal cells were neuronal stem cells, which were isolated from dentate gyrase of the mice brain. So these cells, we could see that morphology changes were not apparent, like they were intact morphology of the cells when we treated with the nanomolecules. Details of this study can be found from our paper, and this is again the sharing of the DNA. So we could find that pilocarpine only treated, the second lane shows the pilocarpine only treated molecule where there is a sharing of DNA, whereas mother molecule as well as the nanoparticle did not show any sharing of the DNA. Also, we found Evans Blue brain study. So Evans Blue stated that MAC and P increase the Evans Blue uptake. That is, MAC and P enters the brain. Once the MAC and P enters the brain, then the volume of the Evans Blue inside the brain increases. So we can find from the graph that MAC and P enters the brain to reduce the effect of the pilocarpine. Similarly, there is also weight of the brain. So weight of the brain increased with the help of, increased when we administer MAC and P. So this suggests that MAC and P has entered the brain. Entry of the brain of any particle or any molecule inside the brain is difficult because of its brain barrier. So, but this particle of this size could enter the brain. We proved it. Then we went for the histopathology analysis, where we found that our particle did not have any prominent lesions on brain, liver, and spleen. These are the three major organs where toxicity happens. So this was not happening with our molecules. We wanted to see the mechanism of action of our molecule. So we went for signaling molecule studies. We employed BCL2, caspase 3, and HSPA1B. We found that caspase 3 activity and caspase 3, all the three signaling molecules were elevated with the pilocarpine alone treated model, whereas when we gave MAC and B, that is a nanoparticle, these signals of these molecules were reduced. These three signaling molecules increased during the neurotoxicity. So coming on to the another cycle analysis, we went for MAP kinase and cytochrome P53 levels. We could find that our molecule has a MAPK kinase and P53 conjugated pathway for its action. This is a biological temp, that is after administering the nanoparticle inside the mice brain by intravenous application. We could find after 24 hours that the sections of the brain showed that there were deposition of the nanoparticles inside the brain. The black dots in E and F are prominent in saying that the those are the nanoparticles which we could find with the help of transmission electron microscope. Now coming to the plan of the experiment, we usually go from normal to cytotoxicity. First we screen antioxidant defense molecules, then we go for inflammation molecules with the help of MAP kinase, NF-kappa B, cytokines, chemokines, etc. Then the tier 3 comes the cytotoxicity, where we go for mitochondrial MTT, apoptosis, necrosis assays to analyze where the toxicity or how effective our molecule is. Few conclusions. This cannot be the end time to decide whether the molecule is okay or not. You have to go for many more tests like ISO 10993, OECD guidance, and anything more for nano products, because nano products do not have a set safety guidelines. So you have to go for a battery of tests to test your safety of the nano molecules which you have prepared. This is my lab, our sir, and this is my team. Thank you.
Thank you, Dr. Gayatri, for that intellectual session we had right now. And with that, as a token of appreciation, I call upon Mrs. Sarita Sadanandan Pillai. Okay, we are passing on to the discussion stage right now before Madam steps down from the stage. The forum is open for discussion. Any questions from the audience? Any questions, audience? So we'll proceed to the, as a token of appreciation, I call upon Mrs. Sarita Sadanandan Pillai, Scientific Officer, Selected Biotech Private Limited, Trivandrum, to present the memento to Dr. Gayatri. I invite all the oral and poster presentation participants to be ready to start the presentations. And now we start with the presentation session oral. And the jury for the poster presentation are Dr. Anish Nair. Jury for oral is Dr. G. Shaktivel, Professor Vimal Karani. I can kindly invite all the juries to be present.
So come to the front.
Once again, reminding all the oral and poster presentation participants to be ready as we start the presentation. We have the jury for poster, Dr. Anish Nair, jury for oral presentation, Dr. G. Shaktivel, Professor Vimal Karani. And we have the first presenter, researcher, Dr. Rishali Ashok Wag, with a paper titled Zinc Oxide Nanobiofertilizers Bioemerging Strategy for Sustainable Agriculture. We welcome you, Dr. Rishali Ashok Wag, as the first presenter for the day.
Yes, okay. Now, initially we collected the soil sample, rhizospheric soil sample. From that, we isolated the 20 different bacterial isolates. And after isolation, we check the metal tolerance assay. In metal tolerance assay, we observe that there are the few bacterial strains which have the capability to tolerate up to the 800 microgram per ml concentration of the zinc oxide, and you can see that key up to the weight, some are responsible to grow, means they are resistant towards the highest concentration of the zinc oxide nanoparticles also. Now, out of these 20 strains, we selected the 3 best strains which are responsible to resist the maximum concentration of the zinc oxide. Then we are taking these 3 isolates and we perform the synthesis of the ZNO nanoparticle synthesis by using this way. You can see the first figure. These flasks are before the ZNO nanoparticle synthesis. After that, addition of the different bacterial strain respectively, you can see the color change after the synthesis of the ZNO nanoparticles by the bacterial strains. And when we extracted it, centrifuged it, and we collected the supernatant, you can see the ZNO nanoparticles which is present in the liquid form, and this is the comparison before and after. Then after that the evaporation will be carried out and we dried it, and in fourth figure you can see that these zinc oxide nanoparticles are present in the dried form. To confirm these particles are nanoparticles, that's why we perform the different analytical methods. In that, first one is the particle size distribution, zeta potential we perform, then FTIR analysis. You can see these 3 bacterial isolates, actually Z1, Z19 and Z23. As compared to Z1 and Z23, Z19 was significantly show the best result, and the particle size is less than 100. It varies in between the 10 to 100. That's why this result is responsible to show that these are the nanoparticles. Second thing, in zeta potential study also, the zeta potential readings are in a negative manner, means this is also one of the supportive result. And third one, we perform the FTR analysis to study the arrangement of the functional groups. That's why we perform the FTR study, and you can see the major peaks which are observed in between 3368 centimeter, that is the OS stretching, presence of the alcoholic group. Second one, 1650 cm, in that it shows the C double bond O stretching. It shows the presence of the primary amide. Third major peak is the 1083 cm. It also shows the CO stretching, means presence of the alcoholic group. And another are also present, 620, 543, 856. This shows the presence of the halogen, BRI and chloride respectively. Now, we selected the one strain Z19 which is responsible to show the maximum nanoparticle synthesis. That's why we selected this one, and 16S rRNA sequencing was carried out of this particular bacterial strain. On the basis of the sequence similarity, this ZN19 was responsible to show the maximum nucleotide homology, and it shows the maximum sequence similarity with that of the Bacillus pumilus. Therefore, this ZN19 ZN19 was the Bacillus pumilus, and you can see the phylogenetic tree also, and this is the sequence data. Next one, optimization study was carried out. Media optimization study was carried out by the statistical analysis, that is Plackett-Berman design. According to this study, there are the three significant factors. Tryptone agitation speed factor is less than 0.01. It 0.05. Therefore, we can say that these are the significant. If the p-value is less than 0.05, then we can say that these are the significant factors which are majorly affects for, or which are majorly responsible for the maximum zinc oxide nanoparticle synthesis. Next, we perform the seed germination assay. Actually, in seed germination assay, we are taking the different concentrations of the zinc oxide nanoparticles, and you can see these seeds have the ability to grow. And these graphs, we actually taken the moon beans. On the moon beans, we performed this particular experiment, and yes, at 10 mg per ml concentration, also these seeds were growing in a good manner as compared to the other one. Yes. Now, next we perform actually the field assay on the watermelon, Citrullus lactus. In first figure, day 1, actually after sowing the seeds, this day 1 represents the day 20 when we apply the zinc oxide nanoparticle, and you can compare the day 1 and day 3. Yes, really these zinc oxide nanoparticles are responsible for the plant growth promotion. And we also check the soil properties. It is also responsible to enhance the major soil nutrients also. And you can see there is the drastic difference. After 1 month, we measured all the parameters, and significantly they are responsible for the plant growth promotion. And we applied it by the trenching method. These are the physicochemical properties of the soil as compared to the soil properties before addition of the zinc oxide nanoparticles. You can see there is the drastic enhance. Nitrogen is reached up to the 144 kg per hectare, phosphorus 16.41, potassium is 349. Means there is a enhancement, or these zinc oxide nanoparticles are responsible to increase the major nutrients of the soil. And most important, these zinc oxide nanoparticles are also responsible to decrease the EC. You can see, as compared to the control, the EC, electrical conductivity, get decreased. Zinc means Zn19 isolate was drastically responsible to reduce the EC, means these nanoparticles are also responsible to decrease the percentage of the salts which is present inside the soil. And you can see here, first graph, it represents the nitrogen and the potassium. Both get enhanced as compared to the control. Yes, here also the Zn19, Zn23, actually three isolates are responsible, but significantly the Z19 Bacillus pumilus was responsible to enhance the major soil nutrients. Hello. Hello. Am I audible? Hello, sir. Hello. We also check the different nutrients, that is zinc, ferrous, manganese. Yes, it also get increased. Electrical conductivity get increased. We check the different parameters of the soil after the addition of the zinc oxide nanoparticles. And recently, we launched the one product, that is the ZN40, which is the nano-based product. We also filed the patent also, and the overall conclusion of this study is that our main objective was that, or our main aim was that, these nano fertilizers are responsible to enhance the plant growth. This ZN19 Bacillus pumilus are synthesized in the large amount. Actually, we synthesize these zinc oxide nanoparticles up to the 500 liter. And yes, the characterization was checked by the particle size distribution, zeta potential, and the FTIR. All three analyses are responsible to show the positive result or the supportive results. Seed germination assay, pot assay. We perform all these assays in a stepwise manner. Seed germination, then pot assay, and then field assay. These also prove that the zinc oxide nanoparticles are responsible to show the significant effect and these are responsible to improve the soil quality also, enhances the major nutrients also, NPK. And yes, the name of this nano biofertilizer is the ZN40, and this nano biofertilizer is applicable for the maize, beans, citrus, sugar cane, onion. Naturally, this is very beneficial for the maize because maize requires the large amount of the zinc. Then other also required cotton, but it requires in a less amount. Beans, citrus, grapes, onion, sugar cane, all these plants requires. And most important, the methods which we perform, techniques, protocols which we use, all are the effective one, economical one, eco-friendly one, and it is versatile one. And these are the some recent achievements.
Sorry to interrupt you, Dr. Vishali Ashok Wag. We are over our 10 minutes now. I would like to remind you, we have a 10-minute presentation. It's 7 minutes of presentation and 3 minutes of discussion. That was a kind reminder.
Hello.
Yeah, go on.
Sir, shall I stop sharing?
Have you finished your
Yes, sir.
Can you hear me?
Sir, your voice is not audible. Hello.
Yeah, can you hear me? Rishani, have you finished your talk?
Hello, sir.
Yeah, can you hear me?
Hello.
Yeah, can you hear me?
Excuse me, ma'am.
Yeah,
Is it audible? Rishali, ma'am? Ma'am, is it audible?
Hello. Hello, sir.
Yeah, hi.
Can
Hello, sir. Hello.
She left. Yeah, I think we can move on to the next talk.
It's real talk.
Hello.
You change the speakers. Third one, yeah.
Hello, ma'am.
Hello.
Hello, ma'am.
No, actually.
Can we just move on to the next speaker? So you just type the message to her saying that we move on to the next talk.
We go on to the next speaker, like B. Grishma Nair, a young researcher, nano-encapsulated. That's a paper title, nano-encapsulated essential oil from Dr. Silicosa induces cell-specific anti-cancer activity through simultaneous induction of apoptosis and necroptosis. A kind reminder that the The presentation will be for 10 minutes, 7 minutes for the presentation and 3 minutes for discussion.
Can you mute them?
Just say hi, man.
Hello, hi. Oh. She can hear.
Yeah.
You can ask questions.
No, we can move on to the next talk. We are running out of time.
So, ma'am, if your session is over, so we are moving on to the next session. Thank you, ma'am. No questions, ma'am. Your presentation was awesome. Thank you.
We welcome B. Grishma Nair onto the stage with the next paper presentation.
Good evening. So myself Grishma and I am from JNT BGRI and my topic of presentation is nano encapsulated essential oil from Totia silicosa induces cell-specific anti-cancer activity through simultaneous inhibition of apoptosis and necroptosis. So breast cancer is the second leading cause of cancer death among women just after skin cancer. And clinically, these are treated by a combination of surgery, chemotherapy, hormone therapy, and radiation therapy, and it all has severe side effects. So most of the conventional drugs that are used to treat breast cancer, they induce apoptotic pathway in cancer cells. But however, the recurrence of cancer and subsequent drug resistance against this apoptotic pathway is exhibited, which necessitate the need for new anti-cancer entities. So Totia silicosa, it's a plant that is belonging to family Aristolocaceae and is a plant of high medicinal value known among the tribals as Kuruvenkantamoli. And they use it to treat a variety of ailments like inflammation, non-healing wounds, etc. And its essential oil is least explored for its pharmacological properties because essential oil, they have low shelf life and high volatility. And all this nano encapsulation of essential oil is an alternate way to circumvent this problem for the development of a stable therapeutic entity because it can provide various advantages like it can provide stability, we can achieve a controlled release, site-specific delivery can be achieved by nano encapsulation. Moving on to the materials, isolation and characterization of Totia silicosa leaf essential oil was followed by nano encapsulation of essential oil in a suitable polymeric matrix and evaluation of cytotoxicity of both the essential oil and nanoparticle on normal and breast cancer cell line. Then acridinone zirconium bromide dual staining and fluorescent microscopy to detect apoptosis. Evaluation of apoptosis by annexin 5 Alexa fluor assay. Then determination of ROS using DCFTA assay and JC1 staining to analyze the mitochondrial membrane potential. So first, this Totia silicosa leaf essential oil was isolated by hydrodistillation in a Clevenger type apparatus and was characterized using GCFID and GCMS. And the characterization revealed the presence of 50 compounds which accounted for 99.68% of the total leaf essential oil. And bicyclogemathrine was found to be the major compound which accounted for 29.33% of the total leaf essential oil. And 37% of the leaf essential oil was monoterpene hydrocarbon and sesquiterpene hydrocarbons. Then nanoparticles were synthesized by method of ionic gelation using chitosan and alginate copolymer system. And a polyelectrolyte complex of chitosan and alginate was formed into which this essential oil got encapsulated. And the particles were characterized using various methods like DLS, theta potential analysis, TEM, and an encapsulation efficiency of 63.2% was obtained, and a stable in vitro release of 68% of this essential oil from matrix was obtained within 24 hours. DLS revealed that the nanoparticle had a size of 631.3 nanometer, which was well within the nano range, and TEM confirmed uniform spherical morphology, and zeta potential of minus 41.2 millivolt was obtained, which revealed that the nanoparticles were relatively stable. And to identify which all compounds got encapsulated into the nanomatrix, GCFID of the nanoparticle was carried out and was compared with that of original oil, and it revealed that most of the essential oil compounds got encapsulated into the nanomatrix. Then MTP assay was carried out to determine the cytotoxicity of essential oil and nanoparticle on normal macrophage, that is raw cell lines, and on human breast cancer cell lines, that is SKBR3. And on treatment with essential oil and nanoparticle, no significant cytotoxicity was exhibited in normal cell lines, that is raw cell lines. But in human breast cancer cell line SKBR3, a significant cytotoxicity was obtained by treatment with essential oil, and a much higher cytotoxicity was obtained on treatment with nanoparticles as compared to the essential oil. Then to analyze how this cell death is happening, apoptosis was evaluated by using etridine on ethidium bromide dual staining, and it was found that on treatment with essential oil and nanoparticle, there was a significant decrease in cell viability, and also membrane blebbing as well as nuclear condensation was observed on treatment with essential oil nanoparticles. So to confirm apoptosis, an HN5 elixir assay was carried out, followed by flow cytometry, and it revealed that after treatment with essential oil, 7.8% of the cells entered into late apoptotic phase. And on treatment with nanoparticle, it was much higher, that is 17.8% of the cells entered into the late apoptotic phase. But interestingly, on treatment with nanoparticle, 8.2% of the cells were exhibiting necroptosis also. So since this necroptosis can be activated by ROS production, ROS was analyzed by using DCFDA, and it was shown that on treatment with nanoparticle, there was a 66% increase in ROS production, that is on SKBR3 cell lines. So since ROS production was also exhibited, a change in mitochondrial membrane potential was anticipated, and that was confirmed by staining with JC1 dye, which can form J aggregates in the mitochondria if the membrane is intact, and that will be lost if the mitochondrial membrane is destabilized. And it was found that on treatment with nanoparticle, there was a decrease in mitochondrial membrane potential. So all the intermediate, that is in apoptotic and necroptotic protein, were quantified by using sandwich ELISA, and it showed that RIPK1, RIPK3, MLKLK, caspase 3, TNF alpha, all were significantly overexpressed on treatment with nanoparticles. So with all these results in mind, we proposed a suitable mechanism that would be happening when the SKBR3 cell lines were treated with our nanoparticle. That is, the nanoparticle, they can either bind to the extracellular receptors or can be intake into the cell by phagocytosis, and the oil will be entering into the mitochondria, and there will be a destabilization of the mitochondrial membrane, culminating in the release of cytochrome C. And the cytochrome C can activate caspase 3, which can lead to fragmentation of DNA, and the cells can be entering into the apoptotic pathway. Along with that, there is an increase in TNF alpha, that was C. That is, this TNF alpha, they can bind to the extracellular TNF alpha receptors, and that can result in polyubiquitination of RIPK1. And this RIPK1 polyubiquitination can, along with the ROS, can lead to overexpression of RIPK3, and both this RIPK1 and RIPK3 can form a complex like necrosome, and that can lead to overexpression of MLKLK. And this MLKLK can cause an influx of sodium ions into the cell, causing the cell to rupture. Yeah.
You're running out of time.
Yeah, I'm concluding. Moving on to conclusion, a novel dual mechanism of apoptosis and necroptosis by nanoencapsulated essential oil from Thodia silitosa on breast cancer is identified. In present scenario, where most of the anti-cancer drugs used for breast cancer treatment follow apoptotic pathway, and a drug resistance is also developing against this apoptosis, this PSNP is a novel phytoentity for cancer treatment. In a nutshell, the mechanism of anti-cancer activity is through ROS and TNF alpha induced activation of RIPK1 pathway of necroptosis, with an augmented classical apoptotic pathway also happening. The limitation in using essential oil in cancer treatment is resolved here by synthesizing nanoparticle which can pave a new avenue for development of new generations of anti-cancer agents. Thank you.
Thank you. Are there any questions?
Just one question. When you check the cytotoxicity against your raw cells, do you check it for the essential oils or do you check it for
Both
the
essential oil and nanoparticles.
both encapsulated one?
Yeah.
Any other questions?
So finally, how are you going to prescribe this as a medicine? Would that be orally or what manner?
Sir, actually this is just an in vitro study.
No, I'm asking what's the plan for the future?
Can we
So how you, right? Yeah, can you bring no problem. So the same result you will get while by the oral?
I think so, sir. Actually, this is just a primary study. We are further we are planning to do animal studies. Sir.
Okay, you did any viscosity study for your oil?
Yes, sir.
Viscosity, spreadability, or your sample, your oil sample?
No.
Not that. Okay. So in future you have to include that.
Yeah.
First you go with the animal study. Basically with the same thing. First you have to check with the toxicity study, then you go for the spreadability and other things. But already I think you need to be concentrating something more, where it's even as a work. Injection and damage is not possible, right?
It's nanoparticle. I think it will be relatively stable if taken molecules. We have to check on that.
Yeah, thank you very much.
And attention for the jury for the poster. Announcement please. The jury for the poster, Dr. Anish Nair and Mr. Anuj E.S. kindly proceed to the poster counters. Also the concerned prospective students kindly be present at the counters together with the jury. Now we proceed to the next session with Atul Hari S. with the paper title Anti-Hyperglycemic Constituents from Ficus Exasperate Wall.
Everyone is going. We need some audience in the hall as well.
Kindly note, we need all the audience be seated in the hall, please.
So that's okay, let's continue.
Those who are not with the poster presentations, kindly be seated in the hall, please.
I'm Madhur Hari from Phytochemistry and Phytopharmacology Division, JNTBJI. Coming to my topic, anti-hyperglycemic constituents from Ficus exasperator 1. So coming to the introduction, diabetes mellitus, a major metabolic disorder characterized by increased glucose levels and rapid lipid and protein metabolism. And its prevalence is more in developing nations compared to developed nations. So development of a new oral hypoglycemic agents are the need of time to combat diabetes mellitus. So for that, we studied the anti-hyperglycemic activity of ficus species. So in traditional medicine, ficus species were used for treat diabetes in various community, various part of ficus species were used. So we screened several ficus species, uninvestigated ficus species for their anti-hyperglycemic activity, and from that data we came to one species called Ficus exasperator. It's called Brahma's bavina, also scientific. So its bark extract shows some prominent activity. So we move on to Ficus exasperator. So coming to materials and methods, the first one is collection. It was collected from our JNTBJI arboretum and it was sequentially extracted with NHC, methanol and water. These extracts were tested for or screened for anti-hyperglycemic activity in through oral glucose tolerance test in overnight pastured Vista. How we done OGTT is that we divided rats into 4 groups, like each cage containing 6 animals, and the control group received 1% between A as the vehicle, and the other 3 groups received respective doses of 3 extracts, and the blood glucose level were monitored continuously after the administration of 3 gram per kg of glucose at 30 minutes after blood glucose. After the administration of glucose 30 minutes, 90 minutes, and 150 minutes. So after finding the potential extract, we carried out activity-guided isolation of these potent extracts to isolate the potent compounds. And finally, we characterized these compounds by using spectroscopic techniques like NMR or mass spectrometry. So coming to the results, positivity of Bioresearch exacerbated stem bark extracts. So from this graph, it's showing the glucose level in these treated animals at various intervals. And from this graph, it's clear that at 90 minutes after the glucose administration, the methanol extract and the hexane extract of exacerbated showed some activity. So we carried out activity-guided extraction of hexane extract and methanol extract to isolate the active components. So coming to the activity-guided isolation of the exacerbated hexane extract, it was carried out through this scheme. So from this scheme, this hexane extract was fractionated using a silica gel chromatography at various stages, and finally we done a reverse Smith column to get an active compound, which is coded as highlighted there. So also we carried out variable doses of this active compound to find the optimum dose, and it was found that the compound show maximum activity at 5 mg per kg of enemy. Okay. So coming to the structural elucidation of this compound. So this compound is a white powder, and it shows a positive result for terpenoids in Lieberman Bouchard test, and also through a GCMS analysis, its M/Z ratio was found to be 297.53. So also we carried out NMR analysis, only one-dimensional NMR, and from this chemical 20 peaks were obtained in C13 NMR, and the chemical shift of 59.4 in C13 and 4.08 in proton NMR indicated the presence of hydroxylated methylene group at C1, and nearby unsaturation to C1 was observed at C2 and C4 in C13 NMR, and overlapped signals at 0.76 and 82 showed 2 methyl groups and peaks at 22.6 and 22.7 further indicate the presence of additional methyl groups. From this data, we came to conclude that the compound is called as a phyto. Also, it is in agreement with the literature data. So first we isolated the phyto. Then coming to the activity-guided isolation of the hexane fraction of methanol extract. It was carried out this scheme, and variable doses were done to find out the optimum dose, and it is found that it is active at 10 mg per kg. So the characterization of this active compound was done to the same way. It is also a white powder, 145 mg, and it showed positive test for Lieberman, which are test for glycosides, and also EIMS M/Z ratio of 577 was obtained. So from this spectral data, chemical shift from 71.8 to 78.78 indicate the substitution other than hydroxyl group at C3, and also peaks from 100 to that 61.53 in C13 NMR were a characteristic of a sugar moiety. Then from this we came to the conclusion that is the molecule has cytosyrol beta D glucoside. So it also agreed with the literature data. Then coming to the activity-guided isolation of exacerbated butanol fraction of methanol extract. This butanol fraction was partitioned with this butanol extraction was we done size resolution chromatography using diion sp20, and after that after doing several reverse Smith columns, we came to an active compound, and it was of 54 mg, and variable doses were done to find the optimum dose, and it was found to be 10 mg per kg. Then coming to the characterization of this active compound, it was a white oily compound, 54 mg, didn't show any characteristic test results. So we carried out this proton NMR, C13 NMR, and CMS. from LCMS data, it was from the M by Z ratio of 478. And from the C13 and proton NMR, it showed 17 peaks, which were corresponding to 3, 4, 5 trimethoxyphenol moiety, and 6, 1 for glucose moiety, and 5 corresponds to RPF ranocin units. Intensity at 56.7 indicated the presence of 2 equivalent methoxyls at C3 and C5 positions, and peak at 59.8 represented a third methoxyl group. From this data, we came to conclude that this molecule has caffeocyl A. The two? You're running over. Okay. So from this, we carried out a comparative data positivity of these compounds, and from this, we find out that common phytol is more active, and also we carried out cytotoxicity assay in fiber cells into MTT assay. Even at 200 microgram per ml concentration, it didn't show much toxicity. Only showed toxicity up to 20%. It's the percentage viability. With this, I'm concluding this session. The 3 active compounds were isolated from this 5-6 aspirator. With this, thank you.
Any questions? Atul, just one quick question. So, did you look at any other anti-diabetic effects besides looking at the glycemic level?
Actually, it's the screening data. We are also doing the further pulse from these molecules.
Okay.
It is under process. We are doing some in vitro assays, and after that we will go for model.
Okay, like what kind of a model?
It's a diabetic model, type 2 diabetes model in rats.
So that's what, like as part of diabetes, like I mean, the glucose is not the only determinant, right?
Yeah.
So you have
yeah, we can go for insulin. Actually, from one of these molecules shows some insulin secretion activity. It was observed in normal beta cells. We cultured normal beta cells, and one of the molecules among these 3 showing insulin secretion activity. But it is we have to do more things to confirm the exactness. That's some process.
Okay. Thank you. Any questions? Okay. Thank you very much.
Okay.
We can move on to the next one.
With that, we move on to the next one, Vidya Bharati S, with the paper title Therapeutic Benefits of Flame Lily Gloriosa Superba in the General and Organ Medicine Review. Kindly welcome Vidya Bharati S as the next presenter. We proceed with the 4th participant of the presentation, Hari Raghava RK, with the paper title Digital Healthcare Management Using Blockchain Technology. We welcome Hari Raghava RK for the presentation. The next one is a group presentation by Hari Raghava and Niveda, and we have Niveda on stage for the presentation presently. The paper title is Digital Healthcare Management Using Blockchain Technology. Kind reminder on the time limitations of 10 minutes, 7 minutes for the presentation and 3 minutes for discussion. Kindly stick to the timing, please.
Good evening, everyone. This is Niveta. I'm from Manaraman Institute of Technology, and this is our group presentation. And here we are presenting our paper on the title Digital Healthcare Management using Blockchain Technology. And the main objective is to implementing digital health science management and grassroots system in COVID-19 using blockchain, which enhance the efficiency, security, and transparency of managing health-related data and during pandemics. So, a short intro of it, COVID-19, which was the pandemic spread in 2019 and 20. It was highly contagious, and we are implementing a new technology for the clinical practice, and needs can be fulfilled through the COVID period. Blockchain technology is the new technology which we are implementing in this blockchain, a distributed platform containing a transaction details. It is mainly for the tracing these medical drugs and health data. According to the literal survey, it is the pandemic is a critical change to the health and safety of the general public, the medical staff, and the medical system worldwide. As the people faces the more difficulties, so in securing of the data and the supplies of medicines and vaccines, it is not available to people properly. The maintenance of patients secure health data is a challenging task for every hospitals in worldwide. For example, in case of main parties, so the data for their security of data was important for us. So for their security purpose, we are implementing a blockchain technology in it. Traceability indicates the tracking of infected patient. It is imperative to control the spread of coronavirus. With blockchain technology, one can track the infected patient's movement, provide real-time data about affected areas, and report direct fighting efforts. This tracing can be done by transaction recording and monitoring capabilities of the blockchain network. Goods and medical supplies need to be tracked to ensure the transparency in a chain. So, blockchain technology can play a vital role in tracking healthcare instruments during the COVID-19 pandemics. Recently, along with health commissions and the economy and IT department, China has launched a blockchain technology-based platform. It consists of recordings and tracking of coronavirus pandemic fighting materials such as masks, gloves, and protection gear. If medical professionals and hospitals had a secure and reliable database of health records, it would reduce the risk of misdiagnosis, and the drugs which we and the medicines which we need to be provide for our patients can be also be detected, and the correct medicines can be given to the patients. So, improvement in clinical supplies by IBM and Boharaja entered a collaboration in 2019 to test the capabilities of blockchain technology in clinical trades management. Blockchain is the best option for supply as it can connect all stakeholders into one supply chain network, universal source, while showing transparency and being able to securely break down data source. The applications it introduces immutability and data provisions while removing single point of failure in the system. We propose a framework along with the algorithm that define the working principles of the proposed proposed blockchain-based tracking system provide a detailed sequence diagram summarizing stakeholders' interactions in the blockchain-based tracing system, tested and validated various scenarios of all system functionalities. Therefore, with blockchain data tracker, any users with the internet can use this secure system. So, as a conclusion, we have presented the utilization of blockchain technology to combat the COVID-19. The critical role of blockchain technology is to help control the spread of this pandemic.
Are there any questions? Yes.
Sir, as we are from
Can you use the mic, please?
So we are from computer science department and we have friends on biotechnologies. They are, when we talk to them, they said that there was an insecure between the data and the supplies of the medicines, so that we had proposed this system for their convenience.
Actually, COVID, this is the only problem of data management in hospital. What
No,
are the other
sir.
biggest management problem there?
Yes, we can. We are having other problems also, but we are on looking through it. We are supposed to see this as a main problem, that the security was first thing for their need.
What I'm saying is, my suggestion is, instead of focus on particular one thing, you just focus on. Did you hear anything about pain care management?
We will go through on it, sir.
Yeah, that's most of in other countries they have the pain care management. So it is common for all those are the people with some normal like spine, they have wound infections, particularly for heart failure, heart attack people. Okay, that was lacked in our places. Mostly in our government hospitals, there is no proper management for that. Actually, this was already submitted by ICMR as a proposal to implement such a database. Okay,
Yes,
for pain care management. It's the same, similar to like that one.
Okay, sir.
That is the most important piece. So in your future aspects, you have to be focused on that one. Okay, that is the most important. That's why I'm asking why you choose this one instead of that one.
Sir, this was mainly chosen on COVID period when we can give a solution. We are thought of this idea. This can be
So
implemented.
what are the data sheets you prepared for this one?
Sir, we are just trying to implement this. We are having a general study on this.
So what are the criteria are going to be implemented in that one? Charge sheet, data sheet. What data are going to be collected?
Sir, the X-rays and what are the medical details we are giving to the patients, and what are all the treatments that the patients are undergone, and what are need to be given. All the details from the top to bottom we will be collected and we can secure through the blockchain. So blockchain is just it can store and secure all the types of data so that we can use this ideas. So for all the treatments that one person undergone can be secured through it.
Okay. Be careful while preparing the chart sheet. Okay. What are the data sheets on particularly?
Okay, sure.
You need to be at least more than 10 queries on that, basic queries. Each queries will have to make 8 queries. Okay, that you have to be very careful.
Okay, sir. Thank you.
Thank you, Divya.
Thank you, sir.
Yeah, we can move on to the next presentation.
Next, we move on to Vidya Bharati S. I call it Vidya Bharati S. The paper title, Therapeutic Benefits of Flame Lily, Gloriosa Superba in the General and Oral Medicine, a review.
present here. I am Divya Bharati, Department of Foods and Nutrition from RBS College of Arts and Science, Coimbatore. I am here to present on the topic therapeutic benefits of flame lily, that is Gloriosa superba in the general and oral medicine. A review article. Gloriosa superba is also known as glory lily, flame lily, or lily climbing or creeping lily. Belongs to the family Colchicaceae. It's native to Africa and also in many parts of tropical Asia, including India, Burma, Malaysia, and Sri Lanka. In India, it is found in Maharashtra, Karnataka, Kerala, and Tamil Nadu and Goa. It is mainly in India, in southern parts, it's mainly grown for its colchicine content. Glory lily grows in red loamy soil with a pH around 6 to 7, and it is mainly commercial, and it's produced mainly for its medicinal properties and pharmacological properties. It is an ornamental plant throughout the world, and it has been used for 1,000 years as a remedy. Due to its over exploitation, it is recorded as endangered medicinal plant. The bioactive component that is present in flame lily is mainly colchicine and colchicoside. These two are alkaloids, and they are known as toxic alkaloids because if they are ingested in high dosage, it leads to fatal condition. Ethnomedicinal uses. It is used as an ethnomedicine in many tribal communities of India. And also in ancient scriptures of India, its properties are mentioned in it. Mainly, it is used for bleeding piles, arthritis, conjunctivitis, asthma, and other many diseases. Pharmacological uses. It is used as anti-arthritic agent because it reduces the inflammation causing cells, and analgesic. It has analgesic properties and uterotonic activity. This extract can be used to reduce the height of contraction during child labor than the contractions produced by the oxytocin hormone. Antimicrobial activity, it mainly act against fungus, that is Candida species, and also against bacteria, Staphylococcus species. It acts as anti-inflammatory agent and also antioxidant agent, scavenging the free radicals. And also anti-cancerous activity, it acts against human cancer liver cells. It has anxiety properties, anti-diabetic properties, and anti-thrombotic properties and anti-fertility properties, where it inhibits the pregnancy in mice, in albino rats during a vivo study. Thank you.
Any questions? So I have a question. So it's a review-based work, right?
Yes,
So how did you conduct this review? Like what methodology was followed and
Methodology, I've collected articles from after 2015 till date, sir. 10 articles I've selected, and from the 10 I have read all the full articles and I have consolidated the points and presented.
so how did you select those 10 articles? Like did you use any kind of inclusion, exclusion criteria?
On random basis, I will select it.
Okay, yeah. Thank you very much.
Thank you, sir. Thank you.
Any questions? Okay, thank you.
Thank you.
Proceed with the next participant, that is Sushri, the paper titled Plant Monitoring System Using Law.
you can announce the orders so that they can be written.
The next participant, Nitish Kumar R, is invited to stage for the next presentation with a paper title Applications of Graphene Quantum Dots in Bioimaging MRI. We have just one more participant for the paper title, and that is Ms. Sushini. Kindly reminding to be ready with the pen drives.
Greetings to one and all. Myself, Nitish Kumar. I am currently pursuing Bachelor's in Biomedical Engineering at Bioresearch Institute of Technology. The paper is about applications of graphene quantum dots in bioimaging. Here is the short view of the paper, characteristics, synthesis, and application, and how it is works in bioimaging. Before we move on to this paper, let's see about how it is prepared. Graphene quantum dots is prepared from the graphene, which is one of the allotrope of carbon, and it has a tremendous amount of properties such as mechanical strength stronger than steel, high loading capacity, and high photostability, high chemical stability, and easily able to interact with the biomolecules and easy to synthesize. Let's see about the synthesis of the graphene quantum dots. It involves in the two methods, whether in top-down approach or down-top approach. Which means the process starts from the bulk material or from the molecular level material. The process involves in physical condensation or discharge or laser ablation, evaporation, irradiation, etc. Here is the exact chemical reaction, what the synthesis occurs. In the bottom up approaches, the small molecules undergoes the dehydration, cell polymerization to result in the polyphenylene, and in the polyphenylene undergoes the carbonization to result in the carbon quantum dots. And moreover, in the top-down approaches, the bulk particles such as carbon sheets or carbon nanotubes undergoes the process arc discharge and electrochemical or physical ablation to result in the graphene oxide fragments. And these graphene oxide fragments undergoes the surface passivation to form the carbon quantum dots. Let's look the eyes on the bioimaging. We all know about the MRI contrast ray, which is used to enhance the quality of the MRI images on the particular areas to see the tumors in the kidney or heart or blood vessels or vein. Particularly see one of the areas. While using this MRI contrast dye, namely gadolinium-based contrast, it results in the mild side effects such as headache, nausea, and itchy skin and cold sensation. Instead of that, we can use the folic acid graphene quantum dots. Since it is the less toxicity, here is a picture of before injection and the post injection. So further applications of the graphene quantum dots are biosensor and bioimaging, photocatalyst, photothermal therapy due to these tremendous properties. That's all.
Thank you. Any questions? So this is basically a literature review, right?
Yeah, yes.
Yeah. So what is going to be your next step? Like, how are you going to kind of like implement this? So you're planning for a study or something?
Sir, we have to check what happens next. We have while we using the polyphasic graphene quantum dots in practice.
Yeah. So how are you going to do that?
Sir, it applies in the less toxicity. So we have applied this folic acid. We have to go to next.
Okay. Thank you.
Thank you, sir.
We have the next participant for the presentation is Ms. Sushini with paper title Plant Monitoring System using log.
Yes, good afternoon everyone. And this is a team presentation. Myself, Sushini. We are going to present upon the topic of a plant monitoring system using IoT. And this topic is of using for finding the moisture level, humidity level, and by checking the whether water content is for a plant is enough or not by using the sensors and by Arduino. And the system can measure the temperature, light intensity of the surroundings, whether the growth of the plant is higher or lower by using technology. And here the input we are using as analog values for connecting the Arduino, and the output will be in digitalized. Here the block methodology for how the moisture and sensor are connected. And the plant monitoring is helpful for watering plants by using the moisture content, humidity level, intensity of the lights, and generally here the plant growth is of we needed mainly thing is of sunlight and oxygen. So here we needed the light content. So for that we are using normally to find the sensors, and here we used soil with the help of soil moisture, and we are placing the small chip-like sensor in the each every plant. This is a basic thing, so we are used in chip type. Uh, and the environmental conditions for favorable to the plant growth is by using the sensors include with the change plant that is related to the change water status includes the snap flow. Here the sensor type we used to porometers or the thermal infrared guns, and the thing which we needed to monitor is of level of plant growth. The system is very used here is for nursery farms or agriculture field. So normally in the agriculture fields, we need to find the growths. Here we are using the other one method of pictures, whether the plant growth is, if it is in dead or is in alive. Here like that we can use the other one method of capturing the image. Here in capturing, we are using the Arduino interfaced camera and processing the image by using image processing to analyze, and the database and retrieval of images. Similarly, in query language, this is providing the appropriate amount of water. In some situation, the whether there is an matrax or in water logging. And here we using a main thing for this is prototype. We using the final values we get in leaves or screen green light is in zero. That small thing zero is of analog values, which is we making analog values into digital values.
Here we use the Arduino code. Main thing is Arduino thing. In Arduino, we are using to code the design what the project we are going to. By using the code technology, we are going to make a small sensor to check the level of water and moisture level. And in future, we can use in same thing pesticides also. If the plant is affected by insects or we need a pesticide for that plant, we can also by checking it. If the plant growth is not there, so we need whether what the pesticides to needed to for the plant, we can use the pesticides. Normally, this is the census makes that it goes the alert message to the owner who handles the form to the mobile connected devices. Is the plant is need of water, that automatically it gets alert to the farmer's mobile. So he can automatically switch on from the mobile. So he can give the connection from the motors and the plants directly. Thank you.
Thank you. Any questions?
What may be the cost for this sensor?
Sir?
Cost, money.
For the small thing, we use just 500 to 600. For small one plant, we just use that.
For one plant, 500.
Yes, sir. But for a total one field, we need it costs a lot.
What are the things you are sensing here? pH, light, pH, light, then
pH, light, the humidity level, moisture, pesticides.
these things comes under one pH meter is there, right? For sensor moisture level, pH level, light sensing level, everything will be in 70 rupees. It's commonly available in Amazon.
Yes, sir. But this now just going on basic thing, I just taken this, sir.
It's a good thing. It's a good initiative thing. What I'm telling is, we just focus on the tissue culture lab needs. Okay. Instead of focus on agriculture needs, because this is not possible to implement for the large agriculture scale.
Yes.
Okay.
For large agriculture, we need a
It's not
simple
possible.
one sensor.
So it is already some these kind of things we already implemented in plant tissue culture lab. We already have these kind of sensors, but they can't be able to sense the moisture level of the tissue culture, tissues inside the bone. I'm not telling about the atmospheric moisture content. I'm just telling about the moisture content of the tissues. Means agar light. That we need to be focused more. That's the most thing we required for profit level. Because we lost more of our money for that one. Okay, we have to be thinking about in that one. Instead of agriculture focus, we just focus on tissue culture lab, because that is also a commercial one. Because now you can see most of you have the aquarium in our house, right?
Yes.
Now everyone turned to the planted aquarium. So all the plant that we are using in our house, it came from the tissue culture lab only. Okay, we are not talking about the vegetable or the things. So if we have focus and if we have some remedies for this tissue culture lab, it will be a good profit for the farmers. Right? So that's it. You need to think about that.
Okay. Thank you.
Thank you. Thank you, audience, and thank you, jury, and thank you, all the presenters. With this, we end the session of the presentation of the oral and the poster. Thank you for all the efforts put in by the presenters and also for the jury. Thank you for having you here. We're happy to have you all here. And now we proceed to the plenary session 2 with Dr. Trupti Patel. There will be an online session with the topic Cancer Diagnosis in and Silico Perspective.
Can I?
Dr. Trupti Patel is a professor, Department of Integrated Biology, School of Biosciences and Technology, VIT, Tamil Nadu. She's an international visiting scholar, virtual teaching, ASFG Developing Country Award winner, and a Fulbright Nehru Academic and Professional Excellence winner, CSIR Travel Grant for International Conference Presentation at European Society of Human Genetics Faculty Summer Fellowship. These are the many accomplishments and awards that she has received. She has a scholarship from Sir Dorabji Tata Trust, and been awarded a gold medal for outstanding scores in MSc Life Sciences. She's an experience of 22 years. We welcome you, Ms. Trupti, Dr. Trupti Patel, to this conference.
Thank you very much. May I start?
Yes,
Thank
ma'am.
you for this lovely introduction. I think I'm hearing my echo. Should I start?
Yes, ma'am, you can start.
Okay, thank you. So, a very good afternoon, good evening, everybody. I'll just start off. This is kind of a basic lecture. It talks about how to, how to use in silico tools for cancer diagnosis and to understand it. So, thank you for giving me this opportunity. And let me just start. Yeah. So we know that there are some complex diseases or conditions like diabetes and cancer and cardiovascular conditions. All of these diseases or conditions do not work in singularity. They work across through combined effects of genetics, environment, and of course the lifestyle factors. When it comes to cancer, what happened was the Human Genome Project got completed somewhere around 2003, and then there are multiple ongoing projects that talk about cancer genome, deciphering the cancer genome. The one of the most important one is the Cancer Genome Atlas program, PCGA. And currently, just 2022, telomere to telomere submission of the whole gapless sequence of human genome was done. So 32 years down the lane, from 2023 on to now, we have now got a complete human genome with every single nucleotide deciphered. Now, what does this help us with? It helps us immensely with understanding the complexity of cancers, and of course, to design proper diagnostics and treatment for different cancers. So, when I talk about cancer diagnosis and therapeutics, here is the interlink. You see that there are certain genes or there are certain variations or mutations to which the genome gets susceptible. When there is a variant susceptible or a specific type of change in the genome, then of course it will change the way the gene will express. So basically, there will be functional changes in the gene expression that may bring about, either we can use those biological insights to understand, clinically understand therapeutics, newer therapeutics, finding new biomarkers, or how to prevent the whole situation. Sometimes we would go back to understanding the etiological processes or the origin of this variant and understand them more clearly, so that we can do therapeutic optimization, or we can go for a proper diagnosis or prognosis, that is after treatment, how would the patient respond to this particular medications that we have optimized. So basically, when we talk about all of this, we talk about biomarkers and drug discovery. Earlier, we were into the discovery of the cancer mutants or the gene mutants in cancer. For what we call it as candidate gene approach. Now, what is this approach? This basic approach is where a particular gene or a set of alleles in a gene would be called as the candidate gene, would be responsible for a particular trait or a particular disease. Okay, we have come a long way. We are today into whole genome sequencing. So we come from candidate gene approach to a whole genome sequencing approach. How does it help? Initially, we knew that recombinant BRCA2 is responsible for hereditary breast and ovarian cancer syndromes. We also thought that developing prostate cancer in a high risk family is up to 40% with specific kind of mutations. Now, this particular estimates could be right when we were looking at candidate approach, But today, when we are looking at the approach of whole genome sequencing, we are able to change this estimate, or we know that we can change this estimate because we know that there is heterogeneity, there is gene environment interaction, and we start taking into account all of this, and we not look for one gene, we are looking for all the genes in the genome. That changes the way we look at the cancer. So basically, from a single gene approach, where we call it as educated gas, we are gone into whole genome scanning, which can, which may be an unbiased way of unveiling the newer genes that would be responsible for a specific type of cancer. Now, just so that you get a basic idea of biomarkers, a molecule that is found in the tissue or the body fluid or blood for that matter, which is a sign of normal or abnormal process, or which is responsible for a condition or a gene. If this particular molecule helps in the diagnosis part, call it a diagnostic marker. If we can target it using specific drugs, we call it a molecular therapeutic marker, etc. So now validating and identifying diagnostic markers, the prognostic markers, predictive markers, or the therapeutic markers. What does it help? It helps in the patient survival, disease management, change the patient outcome. That is basically, give a little longer life than a normal patient would live through all of this. So we want to work on markers. Now, these are certain markers, protein markers that are used in diagnostics, cancer diagnostics, and I just kept this slide for you to understand. But when we want to take a computational approach to study the potential markers, search for potential markers, how would we go about it? So you have the high throughput sequencing technologies. We may mine the data from the available sources, or we may generate a new data. After that, we go for understanding the somatic mutations from the whole genome. There are multiple different types of mutations, nucleotide variants, details that social variations, copy number variation, structural variants, gene fusions, etc. We use multiple in silico tools to call out these variants or to understand these variants. We list out the number of genes that would be involved, or that would be mutating or changing their gene expression, or a mutation that caused them to silence or overexpress. We list out all of these genes, and then we will filter out them from the other genes, saying that okay, this particular genes are recurrently getting altered in a particular cancer, or particular subtype of cancer, particular type of cancer, or multiple pan cancer, that is across multiple cancers. Okay? And this is called as mutational profiling. Once we have done this mutational profiling, we construct or predict novel cancer pathways that would be involved in changing the progression, how the disease progresses, how it initiates, how it becomes aggressive, how it relaxes. Now, and these markers can also become the target for natural drugs or for drug therapeutics. Okay, so all of this can be done by lab. But remember one thing: the final step is validation through experimental wet lab experiments. So here are certain tools. I do not want to go in the detail of it. Somewhere we know the pathways which are changing because of the mutations of biomarkers or the candidate genes. Then we go for David and enrich R, etc. Network-based analysis, which allows the driver mutations to be, seen across different types of cancers or different subtypes of cancer. You can have a comment which will, generate gene alterations across different subtypes again. But this is de novo, that is a novel pathway. This would be a novel pathway that can alter because of cancer. And then there are multiple networks, network plus pathway analysis that combine together. So we have multiple tools, and I would say that this 2 slides will not do the justice to that. Again, if I go through the list of cancer databases, we have a very long list. What I prefer to work on usually is cosmic, and sometimes we use a cancer microarray based database on mine. It's a Thermo Fisher. Again, you have to buy certain data. While Cosmic is Cancer is also one which is available, for free. So we use multiple databases. Now I'm going to talk about some research that we have done. Please do not copy or take a photograph of the slide. The big data is unpublished. We'll search for molecular markers in hematologic cancers or malignancies. How we did that was very interesting. First of all, differentially expressed genes are very important. What are differentially expressed genes? The genes that should be expressed in a certain way at a certain level. Their level either increases, they get upregulated or downregulated depending, on what mutations has occurred. And this particular change in the gene expression is called as differential gene expression or differentially expressed genes. This could be different in different cancers, and they give a lot of information to the scientists and clinicians so that we can unveil certain oncogenic mechanisms. We use multiple pathways like the Illumina data sets, or we can use the microarray data sets, we can use the miRNA data sets, whatever data sets are important and available, and we fix a criteria and then we search for the data sets. The data sets, once we have searched for those, we will start doing a computational analysis to, here remember, our search is like a needle in a haystack. We are searching for specific biomarkers that are altered in hematologic malignancies, in blood cancers, different types of blood cancers. We are trying to find a common signature, and this common signature we are trying to find through how they are affecting the mismatch repair. Coming to that in a moment. So here we use the gene expression of omnibus online Spring David, and we use EnrichR, and we use a lot of doc tools. Now coming to what is mismatch repair, what we did was how these mutations in the blood cancers, different types of blood cancer profiles, affect the repair mechanism, DNA repair mechanisms. We have multiple different types of DNA repair mechanisms, homologous combination, homologous adjoining, nuclear excision repair, base excision repair. But the favorite one that we talk about is mismatch repair. It consists of 9 different proteins, and each of these proteins work in a way to first detect a mismatch in the genome. So it's a replicative error. During replication, by mistake, a wrong nucleotide is added. As soon as it is added, these two protein dimatic complex will detect them. Then there are the other protein complexes which will will be recruited or will be called at that position. Okay, now these 4 protein complexes will now work together, ternary complex, so that they can call the endonuclease, remove the abnormal region where there is a mismatch, use the DNA polymerase ligase and fix it by adding a right nucleotide. So here it was C. Now, because of the mismatch repair proteins, which are 9 different proteins, we get a right nucleotide. And this will happen more often than you think. Every now and then, mismatch alterations occur. Besides this, the 9 proteins are also involved in recombination, meiotic crossovers, antibody diversification, etc. also. Now, having understood that we are talking about a lot of blood cancer cell lines and research done on them, or blood cancer tissues and their gene expression, and how they affect this repair mechanism. We are talking about that. So we collected the data sets from GeoMimas. We gave specific criteria, got 4 or 5 profiles. Then we processed through statistical available GeO2, and then we found log FC. What is log FC? Log fold change. So if the gene expression changes about 2 times more than what it should be, it is called as upregulated. If it is changing and reducing 2 times than what it should be, it is called as downregulated. So log FC will decide whether a particular protein is expressed more than what it should or less than what it should in the cancer. Now remember, in cancer, the gene expression will change because of the mutations, but we are right now not looking at mutational profiling, we are looking at the genes. Okay. Then what we did was we took all the profiles and we found the common genes that were involved in all of these profiles. Okay. Then we constructed what we call it as a protein-protein interactive network using string, and I'll show you that how these 5, 6, 7 proteins which are upregulated or downregulated affect the mismatch repair and its functioning. Okay, the function of the repair mechanism will be, affected. And what happens if the repair mechanism is affected? Wrong nucleotides will spread across the genome, and that will cause the genome to become unstable and will increase more, become cancer will become more and more aggressive. The cancer cells will become more and more relaxed in nature. So we want to stop that. Then we saw that each of these differentially expressed protein, or the gene that we call it, would be would be involved in some pathway, right? Either the pathway is known or not known. So here we are looking at pathway enrichment, seeing whether the genes are important in the cancer, are they contributing to the cancer, upregulation and downregulation of these genes, overexpression and downexpression of this gene. Is it causing the cancer to become more aggressive? We're trying to do that. And after that, we did a lot of docking, and we used some natural product also to check whether we can stop this gene expression from going out of control. Okay. So here is what we did. We took around 6 profiles of different types of cancer, and then we, we found that 846 genes from all of the total profiles. We found 846 genes were either upregulated, the red shows upregulated, the green shows downregulated. Gray is the area where the genes are not changed, they are expressed. Okay, so we found about 846 genes whose expression is changed in the different types of blood cancers. From that we took we did a Venn diagram, and this is how the Venn diagram would look like. It is a tool again. We used the Slico tool, and we found that 30 different genes were found to be common across all of this the 6 profiles that we used. Out of this 30 genes, we still narrowed down. So we said myeloid cancers, chronic myeloid leukemia, acute myeloid leukemia, B cell lymphoblastic leukemia, and B cell lymphoblastic leukemia, Hodgkin's lymphoma, and non-Hodgkin's lymphoma. So we divided them into the as per the cell that they emerge from, the cancer emerges from. So when we did this through second wave, we found that there are total 12 different differentially expressed genes. These 12 genes were differentially expressed across multiple different cancers. Blood cancers, again, we are talking only about hematologic malignancies, and they affected the mismatch repair genes. So we remember that we had 9 different mismatch repair proteins, and of this, not all are involved. Okay, so some is involved with one, the other is involved with something like this, and this is how in silico protein interactome looks like. It is a string, and we see that the black ones are the differentially expressed genes, while the blue ones are the repair genes. So we know that certain genes directly interact with the repair genes, and because they are upregulated or downregulated, the repair gene will be affected. The function of the DNA repair will be affected. So having understood that, we wanted to see that all these 12 genes that we found, DEGs means differentially expressed genes. Once we understood that yes, they are involved in mismatch repair, yes, they are involved in cancer, which pathways they are involved. So first of all, let us, in the normal condition, what are the biological processes, molecular functions, or the cellular processes or components in which they are involved. So these 12 genes were found to be involved in different, they were involved in DNA damage repair, they were involved in double-stranded break repair, they were involved in supercoiling of the DNA binding, they were involved in DNA binding. You see molecular functions and biological processes. Where they were found in the chromosome, the nucleolus, nuclear lumen, they were found in peroxisome, some of the genes. So each gene had some function in the biological processes, in the molecular processes, and was involved in some way involved with DNA repair or with helping the genome to be integrated. Okay, now if these genes have got mutated or upregulated, downregulated, they definitely will affect the DNA repair mechanism. That is what we get from this. But this is not all. We also looked into what are the common diseases in which these genes are involved. So we find, see, cancer, lung cancer, the lymphoid leukemia, Bloom syndrome. Okay, so we find that these genes actually are involved in different types of cancer. Okay, through this two tools which are called as Bailey and Henry-Charp. We used another pathway enrichment analysis, which is called as the National Cancer Institute Nature. That is basically only to deal with cancer. So we found that yes, caspase, FLT3, PTEN, all of this have a role to play in cancer. They get mutated in cancer. They change their expression in different types of cancer. Very important understanding that yes, these genes I did not take. I just took them and I said, okay, upregulated, downregulated, and we see whether they are involved in repair. Yes, they are involved in modulating the repair system, and yes, they are directly involved in cancer. So we found a stronger proof to what we started off with. Then we said, okay, we say that it is involved in different pathways and all. How do we actually see a pathway? So we go through what is called a STAG, and when we went through the CAG pathway, we found that see, our genes are actually present, and the pathway is P53 signaling pathway, very important cancer pathway, inflammatory pathway. If inflammation is there, then also cytokine pathways. Again, we found some of the genes involved. We have 12 genes, remember? Or we can call proteins. Again, they were involved in PI3K pathway. PI3K signaling pathway is an important proliferating pathway. That is, it pushes the cells towards cell division. Higher cellular proliferation, especially the cancer cells with mutations will follow the PI3K pathway and go through a lot of cellular proliferation. So all in all, we understood that there are four, genes, differentially expressed genes, ATM, BRCA1, MYC, and caspase-3, which were directly involved in the mismatch repair. And then there were the certain other genes, and its expression, changing its expression, led to change in the mismatch repair, but via other intermediate candidate genes. Okay, or intermediate proteins. So basically, these are direct, these are indirect genes that were interacting with mismatch repair. Now, we took the four, so this particular work shows that yes, BRCA1, BRCA2, MYC, and ATM and caspase were involved with MSH2, MSH6, MMH1, three different mismatch proteins. This we found through in silico research. We have to validate this, all of this work into experimental research. Now we took this particular upregulated gene. So before I talk about that, there are certain natural compounds that are available. We have in India have been using traditional medicines like Siddha, Ayurveda, etc., homeopathy for a very long time. Okay, and we have been using spices in our food. We use a lot of spices in our food, right? So these spices have helped us a long way in protecting against inflammation, diabetes, this, that. I have used one spice called as curcuma, and a specific compound named compound 3 from that spice to see what happens. Do we change the upregulated proteins here? Do we change their expression so that it can protect us from cancer, or from progressing the cancer very fast? Okay. Now ATRA is on all trans-retinoic acid, also called as vitamin A. ATRA has been already used in cancer treatment. Okay, acute myeloid leukemia is one type of blood cancer in which patients are given ATRA, and it works as a treatment. Now we are saying that there is a compound 3 of curcuma which may be having some ATRA-like properties. So we took the 4 only overexpressed genes, FLT3, MYC, RAD52 and HDAC. What we did was we took their crystal structures of these proteins from the protein database, another database of protein PDB, and wherever we did not have structure, we modeled the structure using in silico tools. Now we got these 4 protein structures, okay, and what we did was we kind of docked them with the ligands. The 2 ligands that we used was compound 3 from curcuma or black turmeric, and we used ATRA. We found that compound 3 could bind easily with FLT3 and MYC. We found that ATRA could easily bind with HDAC9 and RAD3. This is how the docking would look like. The major amino acid here, the shortest minimum hydrogen bound distance was found to be glutamine, asparagine, lysine and lysine, 2 different positions of lysine. Here, this is bound C3, compound 3 binds to these 2 proteins, FLT3 and MYC. HDAC and RAD53 had a more affinity towards ADRA. So some were binding more with ADRA, the other was binding more with compound 3. We found that this particular protein ligand docking helped us to understand that yes, this particular compounds bind to the overexpressed genes. If they bind to the overexpressed genes, they can tune or downregulate the overexpression of these genes. Now remember, we don't want to eliminate the genes or the proteins. We don't want them to completely degrade because they're an important part of our cell. So what we want to do is downregulate them. So we see that the docking is a strong docking, and that means that the overexpressed genes could be modulated. Could be modulated. I'm not saying are modulated. They're still talking about in silico work, right? So ATRA has been already shown to downregulate MYC. And remember, we have MYC here, and it was found to be downregulated by compound 3 also. So what we have, why we want to use compound 3 instead of ATRA? Because compound 3 is, first of all, it is present in the spice. And this particular spice we are using day in, day out. Okay. Second, what has happened is that all trans-retinoic acid has a very short half-life. So despite it being an anti-cancer drug, it's a molecule that we use as anti-cancer medicine. What is happening is because it stays very little in our body, it is draining out of our body very soon. It is getting excreted from the body. Again and again, we have to give large doses of toxic molecules. ATRA is very toxic. It can cause blindness also. So we are looking for different compounds. So we look for compound 3 here. What happened after this is that what I can suggest is that ATRA is good, compound 3 is good. All of these are down-regulating the overexpressed differentially expressed proteins or genes. If this particular differentially expressed proteins are getting down-regulated, then of course they are going to change the way they work on the repair mechanism. That means probably on this repair mechanism, they will work more strongly. That means they will allow the repair to happen, which means that the genomic integrity will be maintained. Okay, genomic instability will not occur. Genomic instability is a major hallmark of cancer. So finally, we say that okay, the conclusion is that high throughput data sequencing spills out a huge data, so interpretation becomes very difficult. In silico analysis helps us to search for right kind of diagnostic and therapeutic biomarkers. Use of natural compounds may protect the irregular expression of the genes. Many genes in silico analysis will reduce the time required for searching the novel pharmaceutically viable anti-cancer molecules. So if you are looking for new therapeutic molecules, we should go first in silico. It will save billions and billions dollars of the pharmaceutical company. Last but not the least, I'll still say all of this study is good. It's in silico. We are validating. We are telling everyone this is possible. But wet lab experiment or experimental technique or wet lab experiments will be required to authenticate, to validate that yes, whatever we did in silico is actually happening. So this is where I would end my lecture. I'll be taking questions if you have.
Thank you, Madam, for that remarkable presentation. A big round of applause from the audience. Thank you, audience. And the forum is open for discussion. Any questions from the audience? Any questions?
Well, either everyone understood all of it.
Are there any questions? Thank you, nothing, ma'am. Thank you.
Okay, thank you so much, and thank you for this opportunity.
Thank you, ma'am. We're glad to have you here today. Thank you. With that, we move on to the next plenary session 3. Ma'am, is it audible?
Yes, ma'am. Am I audible?
Yes, yes.
Yes.
And here we have Dr. Ovia RP, Assistant Professor, Department of Biotechnology, School of Bio and Chemical Engineering, Satyabama Institute of Science and Technology.
Presentation, ma'am.
Pardon me, ma'am. We're going on with the introduction, ma'am, of Dr. Ovia RP, and we are going with the topic, role of mitochondrial ribosomal small subunit protein, MRPs S23 in breast cancer. She is a researcher come academician with a solid foundation in molecular oncology, proteomics, and microbiology, having published research articles in international peer-reviewed journals, recipient of research fellowships from the Council of Scientific and Industrial Research, and DST SERB funded Project India. Open to work with research and development organizations in biotechnology. Ma'am, we invite you to the conference with the present topic. Thank you. Are we audible, ma'am? Hello?
Yes, ma'am.
Yeah.
Sure.
Shall we proceed?
Please start.
Thank you.
Yes, am I visible, ma'am?
Yes, ma'am, you are visible.
Good evening. Good evening to one of our guests here.
Ma'am,
And
can you switch on the screen share? Just switch on the slideshow.
Yeah. Is it visible, ma'am?
No, ma'am, it is not in the slideshow. Just press F5. Just share
How is it?
slideshow, please.
Now is it fine, ma'am?
Yes, just put it in the slideshow, ma'am. Slideshow.
Yeah, it is in slideshow actually.
When it is not showing.
Yes, please wait, ma'am. Now is it fine, ma'am?
Yes, ma'am. Yes, ma'am. You can proceed.
Yes. Good evening. I'm Dr. Odia. I'm assistant professor from the Department of Biotechnology, Satyabama Institute of Science and Technology. Here I am here to present on role of mitochondrial ribosomal small subunit protein MRPS23 in breast cancer. So basically, in mitochondria, there is around 1,500 proteins, whereas only 13 are coded by the mitochondrial genome. The mitochondrial genome works as a separate entity, and here it is specifically functioning for production of 13 essential proteins that are specifically working for OXPHOS, that is electron transport chain, oxidative phosphorylation process, and hence mitoribosome is very unique and it is similar to bacterial ribosomes and it is a 50S particle. Here you have both large subunit and small subunit ribosomal protein. In small subunit you have around 30 proteins. Where my work was related with small subunit protein, and these proteins are like it's a 13 protein complex. It complexes together and it has a distinct, each protein has distinct functions. In this 13 proteins, you have some of them that are homologs to equally, while some of them are specific to only mammalian microribosomes. It's because the eukaryotic form, where the mitochondrial ribosomes has recruited some of the proteins from the other extracellular species. And hence, another 3 proteins that are identified are more specific to microribosomes that are not present anywhere, and these are having extra ribosomal functions also. It's called S37, S38, and S39. So these 3 proteins were discovered in some other extracellular compartment to be as CH1, PTCD3, and then it was assigned for its role as ribosomal proteins. Similarly, in this protein complex, that is 13 protein complex, some of them are known to have other subcellular localizations, and this might have some role in particularly diseases such as inflammatory disease, neurodegenerative disease, cancer. So here we will look into what are the extra ribosomal functions. That is, apart from being a structural component in a ribosome, what are all its other functions? So if we look into these mitoribosomal proteins, when it's an important structural component of ribosome, but we still find it in other extracellular spaces. Here you can see some of them are present in the cytoplasm, in the nucleus, and some are even in the plasma. So definitely it has some other important role to play with. Here I have given an example, MRPS 18B, where 18B competes with E2F1 transcription factor and it also regulates cell cycle entry. So it's one of the important process for tumorigenesis. Similarly, we are looking into what are the extraribosomal functions of these MRPS family of proteins. In my research work, I first focused only on MRPS 6 and 23 based on the literature survey, where MRPS 6 is involved as a neurodegenerative disease, where it is reported as a candidate marker for Parkinson's disease. And hence we also looked into how it is and what is its function in case of cancer. And in case of MRPS 23, previous reports have already suggested that it is overexpressed in breast and uterine cancer, and it is a luminal, particularly luminal type of breast cancer. It is being served as a tumorigenic driver. So in my study, I looked into what is its expression status and what is its functional role in breast cancer. Here we can see the mRNA expression levels of MRPS 23 in various cancers. When compared to HACCAT and HPL control cells, we can see overexpression of MRPH23 in cancer cell lines. However, this overexpression is hormone receptor specific, where we can see ER positive MCF7 or ZR751 having higher expression compared to triple negative breast cancers. And hence, based on these data, we also know that it is having a particular role in one type of breast cancer, or it is having an opposite role in other types that we still get to study. Here, I have also looked into its expression status in breast tumor tissues, where this tissue is treatment name and it is collected by stage 2 or stage 3, and the biopsy samples were taken and Western blot analysis were performed. We can see that it is majorly upregulated in tumor samples when compared to paired normal tissue, that is adjacent tissue present in the same patient. When we compare it, we can see overexpression, and we surprisingly found a second band in our target, which shows that these are being expressed as isoforms, where the lower isoform, that is 17 kilodalton, around 17, this lower isoform is overexpressed in case of cancer cells. And when we looked into the UAlpine database, that is this GNCPTAC database also, we can see overexpression of MRPH23 in breast cancer, and we looked whether it has any effect on survival curve. We can see that there is no significant changes in the survival, however, but still it has some other role, like in case of tumorigenesis, it might play some other role. So we look whether this overexpression has anything to do with the mitochondrial metabolism, whether overexpression of MRPH23 is affecting the production of proteins that are essential for oxidative phosphorylation, that is energy production. So we can see this overexpression and correlation with the expression of COX2. We can see there is no particular correlation between these two, whereas COX2 is also upregulated in the tumor cells. When we compare both, we can see that some of the samples are having higher expression in the tumor and some of them in the paired normal, but still we don't find a correlation between expression of these two. This indicates that mitochondrial metabolism alone is not responsible, while the overexpression has to do something with its extra ribosomal pattern. Then to know precisely what its role in breast cancer, we did siRNA mediated knockdown, and in breast cell line MCF7, we did siRNA mediated knockdown and found and confirmed whether the knockdown is specific. Once confirming, we looked into gene expression of particular EMT markers, some of the oncogenes and tumor suppressor genes. In summary, we can see that majority of the oncogenes are downregulated upon knockdown of MRPH23. Example, LOX, IGF1, EZH2, MYC, whereas tumor suppressor LMNA, TGF-br2 got upregulated in case of MRPH23 mediated knockdown. This indicates that it has something to do with increasing or decreasing other oncogenes and tumor suppressor genes, modulating its expression, and also the EMT. We can find significant difference in ACTP, keratin, ecadherin, vimentin, which indicates that it is also modulating the EMT transition. While knockdown cells have inhibited this EMT transition, and definitely it is having an important role to play in tumorigenesis. To further identify how the role, like other than its important physical interaction with mitoribosomal proteins, we wanted to find whether other ribosomal proteins that are interacting with MRPH23. Then we cloned MRPH23 as a GST fusion tag. Like the GST tag is a high molecular weight tag where we can get highly soluble proteins. In bacterial system, we used it for cloning, and we obtained the pure GST MRPH23 and then performed LCMS analysis. Like we did GST pull down assay where we used MRPH23 as the prey, and the prey proteins here were was normal HEC293 cells, where to find normal functional role of S23, we used normal cell line HEC293. We used those as spray protein, incubated with it, and then tried to identify its interacting partners. Once we performed GST pull down assay, the pull down samples was purified and then loaded onto LCMS. So in LCMS, we were able to identify around 14 interacting partners, where 12 were similar with GST, that is the GST alone control, and some of them were present unique to this MR-based method, where the significant proteins were alone taken into account. We did in replicates to make sure that the physical partners that we obtained were confirmed. So we did in duplicates, and we also checked whether these peptides, the peptides that are detected by LCMS, have significant mass transfer. So finally, we obtained 14 physically interacting proteins with MRPH23, and in that we surprisingly found two important kinases. One is CDK11 kinase, another one is RIPK3 kinase. While CDK11 kinase plays an important role in cell cycle regulation, and RIPK3 is an important protein in necrosis. So we further focused on its interaction with kinase protein, where CDK11 is particularly unique compared to other CDKs, where CDK11 contains two isoforms, one is P110 and other is P58, while P110 is involved in RNA splicing and other important functions, while P58 is particularly overexpressed during G2/M phase of the cell cycle. This indicates that this overexpression of this isoform during cell cycle might play an important role in cancer cell proliferation also. So we checked whether MRPH23 CDK11A, the result that we obtained by LCMS analysis, is further confirmed by in vitro study. So using in vitro analysis, we performed the assays using MCF7 and HEC293 cell lines. We can find that since we don't have a particular antibody for CDK11 P58 isoform, we cloned this peptide, this protein separately, and we also performed the pull down assay. To start with, we performed pull down using HEC293, using MRPH23, and also reverse pull down using CDK11 beads. We found that there is presence of the pull down partner in both the pull downs in both reverse also. And we also done a native pull down. In the native pull down, also we can find presence of particular CDK11 P58 isoform. So to check what is its effect on P58 alone, we overexpress that as in a PCDNA flag tag vector in eukaryotic system. In the CDK11 overexpressing cell line was taken, using beads we did the pull down to find whether MRPH23 is present in this. So obviously we also found that MRPH23 is present in the pull down, in both the duplicate classes, further confirming that MRPH23 is indeed interacting with P58 isoform. To check whether, since MRPH23 is localized in mitochondria, we check where all it is present, what is its subcellular localization. So when we looked into it, we can see MRPH23 is present in both cytoplasmic and nuclear extract, other than its presence in mitochondria. So we specifically isolated nuclear extract, and in nuclear extract, we checked whether this interaction is feasible. Why? Because if the interaction is occurring in nucleus, definitely it has a role to play with transcription or any other important functions that is occurring inside nucleus. And hence, we took the nuclear fraction and performed the pull down. We can find that in the nuclear fraction also there is presence of MRPH23 and CDK11 P58 in the pull down fraction. So further, we also checked by bioinformatics on how this, what is its role. Whether CDK kinase is since it's a kinase enzyme, whether it is phosphorylating MRPH23 at any site. So we know this interaction is occurring in nucleus, and hence we check whether whether there is any phosphorylated sites in MRPH21. So when we checked into kinexes and phosphonet database, we can see serine 11 is phosphorylated, and GPS prediction system also confirmed the phosphorylation of serine 11 residue by CDK kinases. So further confirming by bioinformatic analysis that CDK11A kinase might phosphorylate MRPH23. So we further checked it using MALDI analysis. We know that interaction is occurring, however, we are not sure whether this is an enzymatic case. So we performed a kinase assay using recombinant CDK11P58 flat tag enzyme that is overexpressed in cell line, and then we performed the kinase assay and loaded onto MALDI. We used a synthetic peptide that is spanning specifically the serine 11 residue, and then perform MALDI analysis. And we can find that serine and the peptide sequence was entered and the search was done for phosphorylated form. We can see particularly serine 11 getting phosphorylated, and this was further confirmed in both the substrate MRPH23 synthetic peptide as well as the whole protein JST MRPH23. In both the peptides, we perform two methods. One is fluorescent method, where we can find increased fluorescence in presence of MRPH23 peptide, and also MALDI analysis, further validating the result. So now we know that phosphorylation is occurring, and especially P58 isoform is doing. So this has to do something during mitosis, especially in the G2 bar M fix. So further, we want to check whether this phosphorylation has a role to play in cell proliferation. So we constructed several mutants for this serine 11 residue. Like one is the wild type we overexpressed. The second one is the peptide spanning 2 to 18 amino acids, where the serine 11 is present, is transformed into alanine, serine 11 alanine, and serine 11 glutamic acid, where glutamic acid mutant S11G mimics phosphorylation, while serine 11 aniline cannot be phosphorylated. And another important one is deletion mutant. That is the first that amino acids spanning the 80 amino acids are being deleted and overexpressed. So we used key mutants to confirm whether this phosphorylation has a role to play in cell proliferation. The mutants were constructed, and then we performed overexpression in HEC290 cells and also in MCF7 cells. Initially, we checked with N-terminal deletion, which is the deletion mutant. We checked whether this deletion mutant has lower cell proliferation. We can find that there is lower cell proliferation in case of deletion mutant when compared to the wild type cells. And further, these cells were also analyzed by flow cytometry, where we can find arrest of cells in the S phase. This also may be due to higher cell population that is being stopped or being processed during G2 bar M phase. And further, we also check whether any EMT changes happens during deletion mutant. We can find that the deletion mutant is having lesser EMT action. We can find there is lesser difference when compared to EMT results in wild type when compared to deletion mutant, further indicating deletion mutant were not able to modulate this process. While we check whether deletion mutant is present in cytoplasmic extract or in the nuclear extract, we can see that deletion mutant is present in nuclear extract in more amounts when compared to cytoplasmic extract. Sorry, the wild type is present in more in nuclear extract when compared to cytoplasmic extract, whereas deletion mutant is almost the same. This indicates that the first 50 amino acid is essential for its subcellular localization to happen inside the nucleus. So further, single amino acid deletion was performed, that is serine 11, serine 11, alanine and glutamic acid mutant, where glutamic acid mimics the wild type, that is phosphorylated form of MRPH21. So once we perform the overexpression by transfection into cell lines, we looked into the cell proliferation in MCF7 cells. We can find increased cell proliferation in MRPH23 mimicking phosphorylation. And this also confirmed by flow cytometry analysis. Further, we check what happens to some of these important EMT marker and oncogenic proteins. We can find increased levels of cyclin B1 and there is modulation in the levels of E-cadherin and biotin. So this has something to do with its role in cell proliferation. So we looked into PA3-AKT protein pathway markers and also pro-BCL, this pro-apoptotic markers. We can find that there is increased expression of phospho-AKT and there is also increased expression of CRAF, P8, phospho-AKT or PDK. All these are markers that are responsible for increased cell proliferation and there is decreased apoptosis also. Where we find increased apoptotic inducing signals in the BCL pathway also. Here also we find a significant difference when compared to serine 11 alanine and serine 11 glutamic acid mutants. We also looked into its subcellular localization. Here also we found that single amino acid substitution where we can find serine 11 glutamic acid to be present in more amounts in nuclear extract when compared to cytoplasmic extract. In cytoplasmic extract, it is almost not detectable, while in nuclear extract, we can see this one single amino acid, that is phosphorylation of serine, has a major role to play in the nuclear extract. So since we don't have a particular antibody for this particular phosphorylated form of MRPH23, we raised polyclonal phospho-MRPH23 using mouse Vistar rats. In Vistar rats, we raised this particular phosphor form of antibody using synthetic peptide, and then we check for the total amounts of phosphorylated form of protein in the native lysine. Well, when we did phospho form of LU, we can find in the phospho LU, we can find presence of this particular protein, that is phosphorylated form of S11. And further, we also check other where total extract it was not detectable, while in LU8 it was highly enriched. And also, if you see the native forms, and we plotted it with phospho-MR-based specific antibody, we can find in cytoplasmic extract it is very lower, whereas the total S23 is however present there, but the cytoplasmic extract it is absent, indicating phosphorylated form of S23 is present particularly in the nuclear extract, which is also validated by nuclear specific markers. So this indicates serine 11 phosphorylation has a role that MRPH is entering the nucleus to perform some other function, especially cell proliferation. Well, we also check the expression levels of P58 and P10 in case of the breast tissues as well as cell lines. This also indicates that P58 is present, like overexpressed in case of some of the breast cell lines, and this is also dependent on the hormonal level, hormonal status of the breast cancer. So finally we check whether this role, this interaction between S23 and CDK can be inhibited. Whereas majority of the protein-protein interactions are being targeted as for treatment of cancer. So we also looked into whether this protein-protein interaction can be targeted by an inhibitor that can cause cell proliferation, inhibition of cell proliferation. So we checked whether CDK1 inhibitor, it's a particular type of indolyle derivative. So we checked whether this CDK1 type is similar to CDK11 type also. So we checked whether this inhibitor has a role, and we also confirmed it along with flavopiridol. Flavopiridol is a commonly and it is well known for having inhibitory role for CDK1, and it is especially in breast cancer. So we check both. In case of siRNA knockdown cells, we can find that when we do knockdown of S23 and treat it with both the drugs, we can find there is increased sensitivity towards the drug. This shows that modulating the levels of MRPH23 will in turn increase the sensitivity towards treatment option. So either this CDK1 inhibitor can be used alone or in combination with other drugs, other commonly used drugs, in order to increase the sensitivity of the treatment. And the same was even confirmed in another two cell lines, that is triple negative MDMB231 and MDMB468, and this also corroborated. We can find in both the cell lines the inhibition of MRKH23 gene sensitized the cells towards CDK1 inhibitor. In summary, in the study we can find MRKH23 is overexpressed in breast cancer, and this overexpression is not concordant and it is not having a role to play with mitochondrial metabolism. And the knockdown of this protein also led to increased expression of tumor suppressors and modulation of EMT. And in conclusion, we can say it is having a pro-tumorigenic function, and with its interaction with P58 isoform, MRPH23 is being found as a substrate for CDK11 kinase, and due to this interaction, we can find there is this interaction is inducing increased cell proliferation. This is also confirmed using PI3-AKT pathway proteins as well as anti-apoptotic pathway proteins. Important proteins were analyzed, and this also concludes its important role. A further kinase assay we also performed using both fluorescent kinase method as well as multi-analysis. Both confirmed that kinase is indeed being phosphorylating at serine 11 residue, and by raising particular antibody for serine 11, we also found that this phosphorylated form is present inside the nuclear cells in both cell lines as well as in breast tumor tissues. And these are the publications that arise out of this research study. And further, we are in the process of performing in vivo analysis to look into how to target this particular protein-protein interaction between S23 and CDK11. Thank you. I'm ready to hear any question. Anybody's interested?
Thank you, ma'am, for this very informative session. We proceed to the forum discussion. Any questions from the audience for Dr. Obia?
Yes, ma'am.
We've just put to an open forum discussion for the audience, ma'am. If there are any questions, I think we can go to the end of the session, ma'am. There's no questions anymore. Anyway, thank you for the session, ma'am. Thanks a lot.
Thank you all.
With that, we come to the end of today's sessions. We hope you enjoyed attending our event as much as we enjoyed putting it together. Before we head out, I would like to thank each and every one who have been here with us today. It's been a pleasure being with all of you. Looking forward to see you all here tomorrow at 9 a.m. to begin our sessions. I wish you all a very good evening. Thank you all.
ഇത് ഓഫാക്കി. ഓക്കെ, ദിസ് വൺ. എടാ നാളത്തെ സ്ട്രീമിങ് ഷെൻഹാസ് അത് എൻ്റെ സ്ട്രീമിൽ കൊടുത്തിട്ട് വരുന്നു സർ. എൻ്റെ
ഈ തൊട്ടാ പോരാ പ്രശ്നം.
അതെ, ഞാൻ ഒരു കാര്യം